Blissfully and joyful day, everyone, especially to our graceful professor, Dr. Nicathea Bidoya. At this moment, we are going to tackle about prenatal development. So kindly listen and pay attention as we are going to discuss with you the process of prenatal development. For my part is the summary of all. So here, prenatal development is defined as the process of growth and development within the womb from fertilization until birth. The process of prenatal development occurs in three main stages. The first 2 weeks after conception are known as the germinal stage. The third through the 8th week is known as the embryonic period. And the time from the 9th week until birth is known as the fetal period. The germinal stage begins at conception, when the sperm and egg cell unite in one of the two fallopian tubes. The fertilized egg is called a zygote. Just a few hours after conception, the single-celled zygote begins making a journey down the fallopian tube to the uterus. The beginning of the 3rd week after conception marks the start of the embryonic period, a time when the mass of cells becomes distinct as a human. Once cell differentiation is mostly complete, the embryo enters the next stage and becomes known as a fetus. The fetal period of prenatal development marks more important changes in the brain. This period of the development begins during the 9th week and lasts until birth. The stage is marked by amazing change and growth. Brain development does not end at birth. A considerable amount of brain development takes place postnatally, including growing in size and volume while changing in structure. The brain quadruples in size between birth and preschool. As children learn and have new experiences, same networks in the brain are strengthened while other connections are pruned. During this development, there are factors affecting the child's growth, brain development, and nutrition. Hazard factors in prenatal development are the teratogens. Example, drugs, alcohol, nicotine, and etc. When engaging into, this would lead to critical, nor it cause chronic maternal illness, certain maternal infections, toxin exposures, and nutritional deficiencies. Risk factors in the prenatal period include pregnancy-related complications, prematurity and low birth weight, and infection exposure during pregnancy or at time of birth. A successful pregnancy is absolutely a miracle, called a child, which begins as simple zygote and becomes a fertilized ovum during the first of these stages, or trimesters, of prenatal development. Many scientists believe that anything that affects the environment of the fetus can have an effect upon development beginning at conception and not at birth. Environment does indeed begin to influence the individual as soon he or she is conceived. The effect of environmental influences varies, therefore, in accordance with the stage of prenatal development in which the environmental factor is encountered, as well as the intensity of the threat.
Good day, ma'am. This is Trisha May Arisgado, and for today's report, I am going to discuss all about prenatal development. So, prenatal development is defined as the process of growth and development within the womb from fertilization until birth. During this process, the zygote grows into an embryo and then a fetus. Prenatal risk factors include chronic maternal illness, certain maternal infections, toxin exposures, and nutritional deficiencies. Risk factors in the prenatal period include pregnancy-related complications, prematurity and low birth weight, and infection exposure during pregnancy or at time of birth. And now, let us tackle about some of the hazards of prenatal development. So I chose the genetic problems. Genetic problems plays a major role in development. In some cases, genetic problems can emerge that may impact both the current and future growth of the developing child in the womb. So the first one is Down syndrome. Down syndrome is also known as trisomy 21. It is the most common genetic anomaly during prenatal development. One Down syndrome is caused by an extra copy of the 21 chromosomes, meaning there are 3 chromosomes instead of the usual 2, and impacts approximately 1 out of every 1,000 infants. Typical features of Down syndrome include flattened facial features, heart defects, and intellectual impairment. The risk of having a child with Down syndrome increases with the maternal age. The second one is inherited diseases. A number of illnesses can be inherited if one or both parents carry a gene for the disease. 2 examples of inherited diseases include sickle cell anemia, cystic fibrosis, and Tay-Sachs disease. Genetic tests can often determine if a parent is a carrier of genes for a specific disease. So can we prevent genetic disorders? Only a mother can prevent genetic disorders in your child. Don't leave it on destiny. Genetic tests for a family planning. Often, it is seen that children suffer from the same diseases that their parents and grandparents have suffered from. If a particular disease runs in the family, the children can inherit, and that can put them at the risk. A genetic disorder is a disorder which occurs in a human body due to irregularity in a person's DNA. That is why we often hear people saying that, "Is it in your genes?" This means the common traits or characteristics or even diseases can pass on to an individual at the time of birth from his parents. Genetic disorders are not curable but can only be prevented, and also it is one of several causes of infant mortality. In fact, 20% of the infant mortality in developed countries is due to genetic disorders. The best option to have a healthy baby in future is to get screened before conception and if possible even before marriage. Sometimes, families are very much aware of the disorders in their genes but they choose to ignore the fact without realizing that the outcomes can be damaging. Each year, many parents are surprised by the birth of a baby with some genetic disease. The carriers are usually healthy people, but when both parents are carriers of a mutation in the same gene, then there is high risk of having an affected child. This problem of genetic disorders is increasing due to the lack of awareness regarding genetic disorders and tests which can screen them.
So now I will be discussing prenatal period. While you might think of child development as something that begins during infancy, the prenatal period is also considered an important part of the development process. So the prenatal development is a time of remarkable change that helps set the stage for future psychological development. The brain develops over the course of the prenatal period, but it will continue to go through more changes during the early years of childhood. The process of prenatal development occurs in 3 main stages. The first 2 weeks after conceptions are known as the germinal stage. The 3rd through the 8th week is known as the embryonic period, and the time from the 9th week until birth is known as the fetal period. So the first stage is germinal stage. It begins at conception when the sperm and egg cell unite in one of the 2 fallopian tubes. The fertilized egg is called a zygote. Just a few hours after conception, the single-cell zygote begins making a journey down the fallopian tube to the uterus. Cell division continues at a rapid rate during the approximately week-long journey from the fallopian tube to uterus wall. The cells develop into what is known as a blastocyst. The blastocyst is made up of three layers, each of which develops into different structures in the body. So the first is ectoderm, which is skin and nervous system, and the second one is endoderm, the digestive and respiratory system, and the last one is mesoderm, which is the muscle and the skeletal system. Finally, the blastocyst arrives at the uterus and attaches to the uterus wall, a process known as implantation. Implantation occurs when the cells nestle into the uterine lining and rupture tiny blood vessels. The connective web of blood vessels and membranes that form between them will provide nourishments for the development being for the next 9 months. Implantation is not always an automatic and surefire process. And the second stage is embryonic stage. At this point, the mass of cells is now known as an embryo. The beginning of the third week after conception marks the start of the embryonic period, a time when the mass of cells becomes distinct as a human. The embryonic stage plays an important role in the development of the brain. Approximately 4 weeks after conception, the neural tube forms. This tube will later develop into the central nervous system, including the spinal cord and brain. The neural tube begins to form along with an area known as the neural plate. The earliest signs of development of the neural tube are the emergence of two ridges that form along each side of the neural plate. And the last stage is fetal stage. Once cell differentiation is mostly complete, the embryo enters the next stage and becomes known as a fetus. The fetal period of prenatal development marks more important changes in the brain. This period of development begins during the 9th week and lasts until birth. This stage is marked by amazing change and growth. The early body systems and structures established in the embryonic stage continue to develop. The neural tube develops into the brain and spinal cord, and neurons continue to form. Once these neurons have formed, they begin to migrate to their correct locations. Synapses, or the connections between neurons, also begin to develop. Between the 9th and 12th week of gestation, reflexes begin to emerge. The fetus begins to make reflexive motions with its arms and legs. During the 3rd month of gestation, the sex organs begin to differentiate. By the end of the month, all parts of the body will be formed. At this point, the fetus weighs around 3 ounces. The fetus continues to grow in both weight and length, although the majority of the physical growth occurs in the later stage of pregnancy. The end of the 3rd month also marks the end of the first trimester of pregnancy. During the 2nd trimester, or months 4 through 6, the heartbeats grow stronger and other body system becomes further developed. Fingernails, hair, eyelashes, and toenails form. Perhaps most noticeable, the fetus increases about 6 times in sizes. A word from Verywell, brain development does not end at birth. A considerable amount of brain development takes place postnatally, including growing in size and volume while changing in structure. The brain quadruples in size between birth and preschool. As children learn and have new experiences, some networks in the brain are strengthened while other connections are pruned.
Good day, Dr. Nikitia Biduya. My name is Mary Rose C. Cuyos, and I was assigned to the last part of the report, which is the conclusion. The end result of a successful pregnancy is that miracle we call a child, which begins as a simple zygote and becomes a fertilized ovum during the first of 3 stages or trimesters of prenatal development. During the 9 months or approximately 266 days of prenatal development, the zygote divides into billions of cells, which eventually become differentiated from one another while new systems and parts become integrated. With increasingly sophisticated technology, the fetus has been studied and is considered to be an active agent in its own development. Many scientists believe that anything that affects the environment of the fetus can have an effect upon development beginning at conception and not at birth. Environment does indeed begin to influence the individual as soon as he or she is conceived. As the zygote undergoes mitosis or cell division, the new cells themselves become part of the mother's environment, and through their particular physical and chemical influence, they guide and control the development of further new cells. Different genes are activated or suppressed in each cell, so that while one group of cells is developing into brain tissue, another is giving rise to the heart, another to the lungs, and another to the skeletal system. Meanwhile, the lump of cells is surrounded by the larger environment of the mother's uterus, and this environment is surrounded by the mother and the world in which she lives. The great maturity of women and uncomplicated pregnancies and give birth to healthy babies, and for many years, it was believed that the baby in the uterus was completely insulated from outside influences. Scientists now know that this is not entirely true. Environmental influences ranging from radioactivity and stress in the outside world to drugs, chemicals, hormones, and viruses in the mother's bloodstream can affect prenatal development. Such threats affects the health of the fetus in several ways. This is because the body organs and parts develop at different speeds and go through definite phases. First, they go through a phase of rapid multiplication in the number of cells, then there is an increase in both the number of cells and in cell size. In the third and final phase of development, cell size continues to increase rapidly, but cell division slows down. When a body part or organ system is growing most rapidly both in cell number and size, this is known as a critical period. If an environment factor such as chemical or virus interferes with growth during critical period, development of that organ system will be permanently affected. The effect of environmental influences varies, therefore, in accordance with the stage of prenatal development in which the environmental factor is encountered, as well as the intensity of the threat. And when we talk about teratogens, the scientific study of congenital abnormalities caused by prenatal environmental influences is known as teratology, from the Greek word teras, meaning marvel or monster, and the environmental agents that produce abnormalities in the developing fetus are called teratogens. And in terms of drugs, chemicals can cause a wide range of congenital abnormalities that account for about 10% of birth defects. The severity of the abnormality depends on the amount of the chemical the mother is exposed to, the developmental stage of the fetus, and the period of time over which the mother's exposure to the chemical takes place. In terms of the smoking and nicotine, cigarette smoking has already been shown to have dire consequences for the smoker, and it can be hazardous for the fetus and the newborn child. Maternal smoking increases the risk of spontaneous abortions, bleeding during pregnancy, premature rupture of the amniotic sac, and fetal deaths and deaths of newborns. Women who smoke during pregnancy give birth to babies who are about 1/2 pound or 225 grams lighter and smaller in all dimensions, for example, length and head circumference, than babies of non-smokers, are born prematurely, and have other health problems. And when we talk about alcohol, the effects of alcohol are almost undisputed. Fetal alcohol syndrome or FAS identified in 1973, is perhaps one of the best known and best documented outcomes of drinking, affecting approximately 1 out of every 750 births. And it is not just the heavy drinker who may place her fetus in danger. It has been found that women having one or more drinks daily were 3 times more likely to miscarry than women who had less than one drink daily. And about the diseases, since the placenta cannot filter out extremely small disease carriers, such as viruses, children can be born with malaria, measles, chickenpox, mumps, syphilis, or other venereal diseases that have been transmitted from the mother. And rubella is the most widespread of the viruses that have a teratogenic effect. If a mother or a pregnant mother contracts rubella in the first 3 months of pregnancy, she is likely to give birth to a child with a congenital abnormality such as heart disease, cataracts, deafness, or mental retardation. Interestingly, there is not a direct relationship between the severity of the disease in the mother and its effect on the fetus. Never before has the importance of prenatal care for the developing child been so apparent. With increases in research and knowledge of what factors can affect the zygote, embryo, and fetus, the likelihood that such factors can have a negative impact on the well-being of the child and the mother is greatly decreased.
Okay, that's all for today. Thank you for listening and also for the time that you spent for watching this video. Again, thank you and God bless us all.